Hi. So, have you ever wondered, like, okay, so if humans have like really active lifestyle, they're really cognitively enriched. So, like, we know that, right? If you live an active lifestyle, you are really like the memory formation is better and everything. So actually, how do you test it? You can't really take some human and cut their brain and test it, right? You cannot do that. So in this case, like I took some, and it has been already tested that the lab animals, like the mice, if you put them into environmental enrichment, like give them some toys and everything, they actually perform better in memory formation and different behavioral tasks. So could epigenetics answer this question? So my topic is completely different, I think, from the other projects. So I'm going to talk about how epigenetics can modulate cognitive function. So the main concept came actually with this H2A.Z, which is like a variant of one of the histone proteins, H2A. And it has been recently found that, okay, this protein can actually turn over throughout the chromatin. Like normally we know that epigenetic modifications are like acetylation or methylation, some chemical modifications. But this H2A.Z, which is like a variant of H2A, can actively turn over throughout the chromatin, and it can regulate gene expression. And also it has been found that H2A.Z is related to memory formation. It's like an epigenetic mark for memory formation. So my hypothesis was, okay, so this H2A.Z or its acetylation, could it be differentially regulated if you put them into this active lifestyle model? So for example, environmental enrichment. So what I did was, so I had two groups of mice, both of them were young. So one of them were standard home cage and another one with lots of toys and everything. So it's really happy environment for them. So I kept them for 4 months, and then I isolated the CA1 region because it's really involved in this particular model. And then I isolated the nuclei and stained them and fact sorted them into nuclei and non-nuclei. And then I took the chromatin from the nuclei and did the ChIP-seq chromatin immunoprecipitation followed by sequencing analysis. And I actually found out that due to enrichment, H2A.Z level is being decreased from the chromatin, and it also corresponds with neurogenesis and different metabolism pathways. So one can easily recapitulate that, okay, if H2A.Z level is due to enrichment, H2A.Z is decreased, and if we put some drugs and maybe if we can decrease the level of H2A.Z, and the person might be really intelligent. So thank you so much for your attention. I welcome to my posters. Thank you.